Well, hi, everybody. I'm David Main, President and CEO of Notch Therapeutics. And I just want to thank ARM and the Meeting on the Med for this invite to give you a bit of background on our company. What really differentiates Notch is that we believe that we have unlocked the ability to produce therapeutic T cells from any source of hematopoietic stem cell at scale. And that word at scale is a very intentional and chosen word. And as I'll describe today, we really believe that reducing our technology to practice will be a game changer for the field of cell therapy. Now, just by way of a little bit of background, Notch was founded in 2018 from technology from the Sunnybrook Research Institute and University of Toronto in Toronto, Canada. And our two scientific founders are Dr. Juan Carlos Zuniga-Fluker and Dr. Peter Zanstra, both of whom are world-recognized leaders in the area of both T-cell developmental immunology and bioengineering. I think something that really speaks to the foundation and the science that we have is the fact that Allogene signed a partnership with us in 2019, really after the company was just getting started, and saw that the promise of this technology to be able to really change the face of cell therapy. And then more recently, we've been able to close an $85 million Series A financing that will help us really advance the company towards clinical development of our own products. We're currently about 35 employees and growing very rapidly, and have right now facilities in Toronto, Vancouver, and have just opened in Seattle, Washington. So just a little bit about where we fit in. We are focused in the area of pluripotent stem cell derived therapeutics. As a company, we're very much focused in oncology. And as you can see here, this is really kind of illustrating the evolution that's taking place in cell therapy today, moving from the traditional adoptive immunotherapy of autologous derived products through to allogeneic, and now to be able to use stem cells as the approach for generating an off-the-shelf product. What Notch has brought to this field is the evolution of how to actually control in a very tuned and precision way the differentiation of hematopoietic stem cells through to mature T cells. Now, the Notch signaling pathway has been known for many decades, and there's been many attempts to harness this pathway to create T cells. But to date, these processes have had some limitations in terms of requiring animal feeder cell lines or a fixed plate system, all of which has either increased the cost of scaling, it increases the variability from a batch-to-batch perspective, or it really limits the ability to control the specific phenotype of cell that you want. The first evolution of Notch's engineered thymic niche is what we call our platform technology, is being able to demonstrate that the ligands and signals that need to be shown to hematopoietic stem cells to differentiate into T cells can be done without the need of animal feeder cell lines. And that is routinely what we do. But it's really been this generation 2 that has really set us on the path we're on today, which is moving away from a fixed plate system and using a suspension-based system using bead technology and bioreactors, where we can literally grow billions of cells at a time and finally control and tune the signals that are being shown through the differentiation process to get the phenotype that we like. Today, we are very much focused on producing CAR T cell products, so T effector cells. We have a strong interest in TCR-based therapeutics as well. We've been able to demonstrate both of these through the changing of the signaling in our engineered thymic niche platform. We also have early evidence of the ability to make T regulatory cells, and we also believe that we'll be able to change the signaling that is used in our platform to produce other cell types, such as NK cells and macrophages. But as you would all expect, as a relatively young company starting out, we are very focused, and we're very focused on T effector cells for therapeutic T cell products in the oncology field today. But we are starting to entertain some business development discussions for other cell types and other applications of the technology. Today, we're already at a scale that we think is surpassing what is being considered by others, or at least without the need for massive investment. And maybe I think this is where we would really try to highlight and differentiate our process, is that it's not that you can't build up factories filled with petri dishes to scale a fixed plate system, but that requires a lot of cost, a lot of manpower, infrastructure, and of course, you'll still be stuck with variability from a batch-to-batch perspective, as well as the ability to fine-tune the system really won't be available. We're already working at the liter scale for certain parts of our process, and we're going to be going even larger scale this year, and really feel that, again, we're going to be in a position that once we're ready for GMP and clinical development, that we'll be thinking about thousands of doses from a single manufacturing run, and this is critical, from a small footprint manufacturing process. Something that may not be more than a few runs of 10-liter batches. And we think this is highly differentiated and something that's quite unprecedented in the cell therapy arena. Now, one of the things I like to highlight is that manufacturing has many impacts on product development. The first one that everyone would just immediately assume is that we're talking about the commercial implications of lower cost of goods, reduced logistics, which may make this therapy overall just much more accessible to patients. But that isn't the part that I like to focus on. I think there's other elements of the improved process that's going to translate into better products for cell therapy. Within the clinical sphere, we're really talking about one cell line for multiple patients. Again, so a lot of homogeneity in how we make our products, tightly controlled, and really envisioning a product that is the same time every time. This is going to really allow different kinds of clinical investigation, such as repeat dosing studies, dose response curves, even thinking about how different phenotypes within the cells may translate into different clinical applications. And at this point, those sort of things have been quite difficult to investigate fully. And we feel that this kind of highly controlled, quality by design manufacturing system is going to open up new clinical avenues of investigation of cell therapy. Also, from a regulatory perspective, the fact that we have no animal components in our system, reducing the need for purification or tracing of animal components in the final product, greatly reducing the variability through tightly controlled manufacturing processes, and a single chassis streamlines the development of multiple products. So I think that regulatory-wise, we will also have some great advantages. So altogether, I think these three areas really, again, as I mentioned, speak to is how this could really start making cell therapy more drug-like and using the same kind of parameters and approaches to development that have been done historically in drug development, and bringing that to the cell therapy space. And we think that that is really a new promise that could really expand the potential for cell therapy. We're already a growing company and have a great access to world-class facilities, and we're building on that. In Toronto, we have access to GMP cell banking and GMP manufacturing facilities, is where we have clinical grade gene editing available to us. And we have a great partnership with the Center for Commercialization of Regenerative Medicine, or CCRM. And in fact, they're a founding partner of Notch. And so it's really much more than just a contract manufacturing relationship. We really are partners, and we're starting to envision how we could grow together and expand what we do together. And in Vancouver, we have our bioengineering and research team and the beginnings of our corporate team. And we've recently just added a facility in Seattle and starting to grow our translational sciences and development expertise in the Seattle, Washington area. Very experienced team across all fronts, from corporate leadership, science leadership, and development of cell therapies. And again, I think that this is something that we're quite proud of in terms of bringing a lot of expertise to the table early on to advance our product development initiatives. We've also surrounded ourselves with good partners. I've already mentioned CCRM, but Allogene as well came in early, have been great partners. We're focused on enabling a number of their targets in the hematologic malignancy field that will be using our off-the-shelf iPSC-based approach. And it was a very attractive financial partnership for us. And we are starting to entertain additional partnerships in areas that we don't intend to pursue ourselves. So we have the early beginnings of a pipeline, but what I like to really emphasize is that what we're trying to do is, rather than to rush a product into the clinic, it's really bringing this quality by design to an entire new approach to how to make cells in a consistent, highly controlled manner that we think will bring a more expeditious approach to our clinical development aspirations and set a new standard for the regulatory requirements for cell therapy and how cell therapies can be viewed as a mainstay therapy being treated just like any other kind of drug in development. Hopefully you'll find that as a very good introduction to Notch, and I look forward to any kind of questions or interaction that we may have. And thank you very much for your time today.